All Neet aspirants, you are listening to podcast Botany for Neet by S. Ramakrishna, Cyclic and Non-Cyclic Photophosphorylation. As photophosphorylation indicates the formation of ATP from ADP and IP in presence of light, that is photophosphorylation. As cyclic and non-cyclic photophosphorylation, all organisms extract the energy from oxidizable compound and stored in the bond energy in chemical bonds of ATP. As ATP it is having 3 phosphate, the bonds between the phosphate having what it is the energy. So stored in chemical bonds of ATP. What is oxidizable substance? In generally oxidizable substances are glucose, NADH and FADH2. Here in case of photosynthesis, which is oxidizable? Water as well as plastophenol PQH2. Later these hydrogens are utilized for the formation of what it is ATP. Next. So formation of ATP in presence of the light is photophosphorylation that occurs in chloroplast, and formation of ATP in mitochondria it is oxidative phosphorylation. In photophosphorylation it is light driven, but in case of oxidative phosphorylation is oxidation reduction. What is oxidation reduction? Oxidation of NADH and FADH2 and reduction of oxygen. But in photosynthesis it is a light driven process. And phosphorylation occurs during non-cyclic movement of electron. What do you call non-cyclic photophosphorylation? During the movement of electron, electrons are not used up. Energy of electrons are used to pump the proton from stroma to lumen. Later, these hydrogens are utilized for the formation of ATP. As electrons do not cycle back to the same chlorophyll, we are calling it as non-cyclic electron transport. And whatever the hydrogen accumulated due to non-cyclic electron transport utilized for the formation of ATP, that's why it is considered as non-cyclic photophosphorylation. This non-cyclic photophosphorylation occurs in case of grana thylakoids. In contrast to this, there is a cyclic photophosphorylation. It operates only when there is only one photosystem is operating, that is photosystem 1. In non-cyclic photophosphorylation, both the PS2, PS1, which is connected by cytochrome. So PS2 in conjunction with PS1 and it is connected by it is the cytochrome complex. In grana thylakoids, in grana, what are present? PS1 present, PS2 present, NADP reductase, that is the enzyme located on outer surface of grana thylakoid membrane facing the stroma. NADP is a coenzyme present in stroma, but NADP reductase is an enzyme present on membranes of grana facing stroma. And these grana membranes having CF0, CF1, that is ATP synthase. So overall, grana having PS1, PS2, NADP reductase, CF0, CF1. And stroma lamellae which connect the two grana in which cyclic photophosphorylation occur. Because primary acceptor of electron now not transferred to NADP because there is no presence of NADP reductase. Electron cycle back to the same chlorophyll. So only ATP formation occur in case of cyclic electron transport. In non-cyclic, ATP as well as NADPH2 formation occurs. In case of stroma lamellae, PS1 present, PS2 absent, NADP reductase absent, CF0, CF1 present. So in both the cases, PS1 common in grana as well as stroma lamellae. CF0, CF1 present in grana as well as stroma lamellae. So ATP formation common for stroma lamellae as well as it is the grana thylakoid membranes. So non-cyclic and cyclic. If you differentiate non-cyclic, cyclic. In non-cyclic, electrons move in zigzag fashion. In cyclic, electrons move in closed circuit. In non-cyclic, both PS1 and PS2. In case of cyclic, only PS1. In non-cyclic, ATP, NADPH2 formation. In case of cyclic, only ATP formation. In non-cyclic, splitting of water occur, but in cyclic, there is no splitting of water. Why there is a splitting of water in case of the grana membrane? Because presence of PS2 physically located on the membranes of thyroid facing what it is the lumen. But in case of the stroma thyroid, there is no presence of PS2, there is no splitting of water, there is no evolution of oxygen. And in case of non-cyclic and it is the cyclic, the differences. So overall, the phosphorylation is non-cyclic photophosphorylation as well as cyclic photophosphorylation. I am signing off from this episode. You will see in case of the next episode, that is chemi-osmotic hypothesis, very important and concept based, and tricky questions are expected from chemi-osmotic hypothesis.